Hello. I'm really excited to be here and to report that I found a fantastic home for the Martinez lab at Stanford. Here we go. And I've had a blast setting up my new lab and spending time with this amazing group of trainees that I've managed to recruit to my lab. And we're really interested in cracking the pre-mRNA modification code in health and disease. So I'll remind you that pre-messenger RNA is transcribed from DNA, and then it is processed by splicing to remove introns and join exons, by cleavage and polyadenylation to make mature messenger RNA that can then be translated into proteins to achieve cellular functions. And over the past decade, it has emerged that messenger RNAs are extensively chemically modified to make non-canonical bases. And my own work and that of others has shown that these non-canonical bases are added actually very early to the pre-messenger RNA and can affect these processing steps, such as splicing, to affect gene expression. And we're really interested in how this component of the central dogma establishes gene expression programs in a variety of cellular conditions. One of the modifications that we study is pseudouridine, which is an isomer of the canonical base uridine. And this isomerization is carried out by these RNA-modifying enzymes called pseudouridine synthases. My lab is studying how mutations in these enzymes cause neurodevelopmental disorders and how upregulation of the enzymes leads to cancer. But in order to understand this, we need to know where these modifications are in mRNAs and what do they do. So mechanistically, we're asking, how are specific mRNAs selected to be modified? Are there readers of these modifications or proteins that can interpret this code? And how do these modified sites affect pre-mRNA processing and other steps of the mRNA life cycle to establish gene expression programs in a variety of conditions? So we're using chemical labeling of RNAs and high-throughput sequencing, biochemistry, and model system to ask these questions. And I'm happy to talk about whether RNA modifications might be playing a role in your systems of interest. Thank you.